Opa, maravilha, vamos lá. Rio, São Paulo, podemos?
Tudo
Maravilha.
ok aqui,
Então,
Joy.
ok, então vamos lá. Hi everyone, we're here for a new edition of the Immuno webinars. Today we have Marco Ataide with us. Marco is a fresh hire for the School of Medicine in Ribeirão Preto, USP. He's graduated in biology at PUC Minas, then did his graduate studies with Ricardo Gavinelli over at UFMG. Worked a long time in Germany at the Max Planck and other institutes, related institutes. Came back to Brazil, worked a little bit with Dario Zamboni in Ribeirão, and then went to MGM at the Bay Area, where he spent the last years working as a PI there. So Marco works a lot with T cell. I think that's the story he's going to tell us, and we're anxious to hear your talk, Marco. Thanks a lot for giving us the pleasure. Okay.
Right. Thank you, Thiago. Thanks for the invite. I'm very happy to be here today. Very briefly before I go, when I went to US to join Amgen as a scientist and a group leader in an industry, I thought, yeah, my career in academia is gone, it's done, I'm done with that. But I was still doing some basic research there. I still have a postdoc at Amgen, and I said, well, I still love the basic research. It's something that I still have to try again. And then I had the chance to join USP in Ribeirão Preto. It was a great moment. And again, I'm very happy to share the story with you guys today. And that story, I mean, it's nice. I mean, this work was published in Nature Immunology back in, I think, end of 2020. But so many things are coming up on top of that story, especially in terms of CAR T cell as a therapy. And I think it deserves to be recounted again, and the story. And when that story was published, it was in the middle of the pandemic. And unfortunately, I didn't have much time to spread the news and how important BATF3 is to T cell biology. And again, I think it's a great opportunity to share that story and what could come after the findings we got back there, and especially because we have some T cell biologists here in this audience, and I hope you guys enjoy it. So yeah, before we start, let's just yeah, I think now I can click. Oh yeah. Yeah, I love that cartoon. This is a quite old. This is from Haf Ahmed publication back in 2003. That was the moment that people started to understand the importance of CD4 help to get a proper memory in terms of CD8 T cell immune response. So you can get a memory CD8 T cells without CD4s, but the amount of cells you get later on is very small, and the quality of those cells is also very poor. So this is a very classical data showing, how CD8 T cells can efficiently or not kill some target cells. This is in vitro in the presence or absence of CD4 T cells. So here in the square, we have the CD8 T cells that got primed in the presence of CD4 T cells, and he's in the absence. And this is during the secondary response, when the cells, CD8 T cells, re-encounters the antigen. And the primary response works fine, but the memory doesn't. So, and then people try to, we understood the importance of CD4 in the priming phase, but when in terms of time and how and where in terms of space in the lymph node this event happens, and that question was out there for a long time, and part of it is still open. But in 2015, a very nice work from Casting Miller and this is a Custom-Miller model, shed some lights on that question. So what they found was that this priming phase is divided in two moments. At the beginning, we have infected DCs presenting antigen to CD8 T cells, and then at the same time, but in a different part of the lymph node, here in the interfollicular area, and then a little deeper in the paracortex, we have non-infected DCs, but of course, with the antigen loaded, presenting the antigen to CD4 T cells. And here you see a very important population called XCL1-DC. We also know that population as CDC1 or CD8 positive dendritic cells. And those cells will be also part of a little bit part of my talk today. And that population is there, and that population actually plays an important role later on in the late phase of the priming. So that population works as that cell type works as a platform where CD8 T cells and CD4 T cell meet. And that's the moment where we think the help happens. And then another study from the same team that complemented this initial work showed that actually CD8 T cells, here represented in red, are central players in the construction of that environment, that microenvironment inside of the lymph node. So CD8 T cells, they produce XCL1, which is the ligand for XCL1, and also CCL3, which is the ligand for the CCR5. And then in that way, CD8 T cell recruits PDCs, both PDCs and XCL1, to that cluster, to the late phase of the priming moment. And then in that way, so the cross presentation is enhanced, and then we have the optimal antigen presentation and the help, and then you get a proper memory formation. So this is just to illustrate how important that initial moment of T cell activation is to get a proper CD8 T cell immune response later on during the memory phase. Okay. So before I tell you a little bit about the BATF3 and how BATF3 controls the function of DCs and late on T cell, let me just quickly remind you about this huge transcription factor family. It's called AP1. This is a general name for this group of molecules that can be subdivided in two different subfamilies. So the ATF, the Jun, FOS, and MAF family. So those factors are everywhere in the organism, in different cell types, not only in immune cells. It controls several cellular functions. And of course, is also important for the immune response. And BATF3 is part of the ATF family, which is a large, actually, subfamily. Just click here. So typically, the AP1 molecules, they form homodimers or heterodimers. So here's just an example of Jun and FOS. That's the most classical AP1 transcription factor, heterodimer. And this binds to a specific region of the DNA and promotes the transcription of genes. And at the beginning, people thought that the BATF and BATF3, most of those ATF family, they were like inhibitory type of transcription factors because they didn't have the DNA binding region. But then later on, we realized that actually those factors, BATF and also BATF3, they can be not they can be, they need to be associated with these interferon regulatory factors, IF4 and 8. In the case of BATF, typically we have it pairing with IRF4 and BATF3 with IRF8. So as I said, in my previous slide, GCs, especially XL1 GCs, are very important players for the priming phase, where you want to get a CD8 T cell memory formation. And back there in 2008, we didn't really have a let's say efficient tool to specifically deplete CD8 alpha positive DCs or CDC1s, which one we now call XCL1 DCs. And in that study, they were looking for genes that could be specifically expressed in DCs. And they have done that screening, and they found BATF3. And then they developed the full knockout BATF3 mouse line. So that mouse line, by the time, didn't show BATF3 in all the cells, including DCs. And because of the deficiency of BATF3, they found that DCs couldn't develop properly. So that mouse line have a huge defect in the CDC1 development, very, very few cells. So then they published that story, showing that BATF3 is the central factor for CDC1 development and function. And then, for example, here is a data from that paper where they transferred these fibrosarcoma cells, so it's a cancer model, into BATF3 knockout mice. And then the control of those cells is highly dependent on T cells, especially CD8 T cells. And then when they compare now the wild type versus the KO, so this is BATF3 KO, we have a huge difference. So the knockout mice, they cannot control. And this is just a control direct to knockout. So believing that without DCs, you're not going to have a proper CD8 T cell function. That was the conclusion. So, right. And again, the question about, what are the signals that CD8 T cells get during the priming phase in the first, let's say, 48 to 72 hours, that controls the function of those cells, or the CD8 T cell memory formation. It was a very interesting question for me back there. And then I start basically doing some single cell sequencing meta-analysis, looking for genes that could be highly expressed, at least in that moment of the immune response. And then I luckily found a gene called BATF3. So here, just to guide you briefly, this is CD8 T cells. This is P14, so CD8 T cells that responds to the LCMV. So this is the virus that you can, have a clone that can induce an acute or a chronic infection. This is the acute infection. This is the clone Armstrong. So all antigen-specific CD8 T cells. And then we have a different time points here to represent the different moments of the CD8 T cell activation, right? The priming, so by day 3, the expansion and effector phase, contraction by day 14, 21, and the memory phase. And interestingly, BATF3 was very sharply expressed on day 3 in a population on day 3. And then it was gone. So then I was thinking about the Kenneth Murph's work, the CDC1 that I just showed you, that they claim that BATF3 is expressed on DCs and control DCs function. But now I've seen BATF3 being expressed in activated CD8 T cells. Probably they missed, if they didn't activate any T cells back there, they probably missed that expression in CD8 T cells. And in the full knockout mouse system, of course, without DCs, you don't mount a proper CD8 T cell function or immune response, but it's very possible that in the system, the non-functional CD8 T cells they have shown was also due to the absence of BATF3 in CD8 T cells, speculating that, this type of expression might play a role. So the cells would not express such a molecule in a very specific time point to do not have any function. That was the idea. So, yeah, so I decided to take a look. This is a single cell, this is meta-analysis. And then I did some experiments. This is just part of it. And this system, in this system, we use the OT1. So this is a in vivo activation of CD8 T cells. And via mRNA, I also found BATF3 being expressed basically at more or less the same time point I've seen in that data set. So it was crystal clear to us at the time that BATF3 could play a role in intrinsically in controlling CD8 T cell function. All right, so and then we've done different type of experiments, and this is let's say one of the killer experiments. So what we decided to do was, again, taking advantage of the OT1 system. As you know, those cells respond to ovalbumin peptide. We transferred wild type OT1s and BATF3 knockout into the same recipient. And then we used this listeria expressing ova to activate the cells. And then we, at day 30, by the memory phase, we challenged those mice with vaccinia, also expressing ova. And then we did the analysis at day 6. So, and here's the data. Let's take a look. And then we compare wild type and BATF3 knockout side by side. So by day 8, when we checked just the relative frequency and to see how the cells look like, we didn't really see any difference in terms of frequency or numbers. Function, those cells, they produce normal amount of cytokines. They just proliferate normally. It was a little bit frustrating, to be honest. But then during the secondary response, as we know, back there, we can mount a memory, a pool of CD8 T cells, but without the proper prime, the secondary response is typically weak. So then what we found was a really dramatic difference comparing the wild type OT1s with the BATF3 knockout. So just got very, very few BATF3 knockout CD8 T cells during the recall. So, and here could ask, so what is the defect? Do we have a smaller memory pool? And that's why let's just go to the next slide. That's why you have a smaller amount of cells during the recall. So is a problem in terms of numbers, or is the quality of the cells? You have the same amount of cells, and now during the recall, the cells they are just behaving poorly compared to the wild type control. It could also be both. That's what we investigated later on. And just to make the very long story short, I'm going to show you one of the experiments that told us that one thing is numbers. So here's the experimental design. We basically caught transfer, as I showed you initially, the wild type and the BATF3 knockout. So by day 7, we don't see much difference. But by day 12, we start seeing some difference. So less BATF3 knockout CD8 T cells compared to the wild type. And then by the memory phase, we have that big difference. So we thought, well, those cells, they are contracting too much. So during the contraction phase, we are losing those cells. That's one answer. So let's see, put those cells together again. So those are the memory right? So we had the naive, and now we have the memory. So what we did, we just gave the cell the same chance to respond. And then we sort those cells, put them again 50-50, and then we transfer those cells into another recipient, and then we infect them again, and then we did the analysis just 6 days later. And again, less pronounced, as we saw, we still see a difference in terms of expansion of those cells. It's important to say that even here, those cells, they do not produce less cytokine. The cells that can survive here or here, so the memory, among the memory pool or the recalled cells, they produce the same amount of cytokine. There's not much defect in cytokine production, but the cells simply cannot proliferate and expand as the wild type. And then with that we conclude that this is a problem, of course, of numbers. The cells, they probably contract too much, but still the ones that survive, when we try to recall them, they cannot proliferate as the wild type counterpart. Yeah, so we, of course, to try to understand what is the problem in terms of quality, we decided to do RNA-seq, and we've seen a huge transcription network, let's say, dysregulated. It was very difficult to pick one molecule. I can tell you, for example, L7 receptor was highly downregulated. Back there we thought, well, BATF3 regulates a huge network. It's not only one molecule. But, as typically for big journals, you have to get one molecule. What is the molecule that, if you hit, you can rescue that deficiency? And then we found one molecule that could be one of those. And that's the molecule called BIM. So, which is a pro-apoptotic factor, right? So we noticed in our RNA-seq that the mitochondrial pathway was upregulated. And then, because of that, we decided to screen for some of those pro-apoptotic molecules. And as I said, BIM is part of this intrinsic pathway, and it can block BCL2, and then when BIM is present, you have an increase of BAX and BAC translocation into the mitochondria, and then more cytochrome C being released, and the chance of the cells to getting apoptosis. And then when we check for BIM expression in the BATF3 knockout CD8 T cells, and here's again the whole time points of the immune response, representing the different phases. What we found was, in steady state, the cells, they have high levels of BIM. And then when they get activated in terms of antigen-specific way, they start down-regulated BIM. But the BATF3 knockout CD8 T cells, they couldn't do it so efficiently. And then by the memory phase, they have very high levels compared to the wild type. So then we thought, well, interesting. We know this is not the only thing happening, but what if we silence BIM in the BATF3 knockout CD8 T cells? And then we used the shRNA system to silence BIM, and then we silenced BIM in the BATF3 knockout CD8 T cells, and then compared them, compared them with the wild type, the scramble control, sorry, and of course we transfer those cells into in vivo system, and then we infect those mice, and then wait for 7 days. So here's what we saw when we compared scramble versus scramble. So we have the wild type scramble and the BATF3 scramble, very much what we've seen. And here's important to say that those cells, they are not naive anymore because we have to activate those cells in vitro. Otherwise, it's very difficult to deliver our shRNA. So that is slightly different from the system we use with naive CD8 T cells. But still, by day 7 post infection, we saw less BATF3 knockout. But as expected, both had scramble. However, when we promote a silence of BIM in the BATF3 knockout CD8 T cells, we could revert the phenotype. So now we get more BATF3 and knockout CD8 T cells when BIM is silenced. And that difference compared to the wild type is not because the wild type cells are doing worse. So if you, for example, compare this wild type scramble with that scramble here, they just do normally. So those cells, they are in the same mouse and those are in the same mouse. The point is here we have a defect in the cells, they cannot expand, and here or die, they are dying more, and here the cells are not dying. So we could, by simply silencing BIM in the BATF3 knockout CD8 T cells, rescue the phenotype. And again, very likely here is just controlling the cell death. Oops, sorry. Just a second. All right, so now there was a loss of function of BIM, but then we thought, well, that would be very interesting if you promote gain of function of BATF3. Can you make those cells behave in a better way? Can we get more memory cells later on? So here's just experimental design, very simple. The same system, we used the OT1 system. And then the OT1 T cells, we transduced those cells with retrovirus, carrying our construct to express BATF3 or the control. And then we activate those cells in vitro and follow the cell culture for a couple of days, or in vivo. And here's the data. So that's the in vitro very quickly, because you cannot keep those cells for many days in vivo. Well, in vitro, you can, but it's just, you just consume tons of resource. But by day 3, we already saw a very big difference. So now don't get confused. This is BATF3 gain of function, not deficiency. And then we just revert the whole thing. We get more BATF3 by that time point. And now when you transfer those cells into in vivo system, it's basically the same thing. Again, those cells, they are not naive, right? Because we activate those cells in vitro. So by day 7 post-infection, we already see a difference, and this is representing the memory phase. So the cells, they really do much better, and now we can increase the memory pool by day 30. And interestingly, when we check BIM expression in a BATF3 gain of function, so let's check here. This is day 4 from the in vitro system, and in blue is the overexpressing the BATF3 overexpressing CD8 T cells. We see lower levels of, so let's check here, of BIM, those cells, correlating quite a lot with the BATF3 loss of function where BIM is upregulated. So then we did so I'm just moving. I don't know if you guys want to ask any question. So please just stop me. But yeah, if not, we can also discuss in the end. But we decided to run a RNA sequencing now in the BATF3 overexpressing cells. And we found, as we found before with the deficiency, a lot of genes being dysregulated or downregulated or upregulated. We see again a big network changing when BATF3 was overexpressed. And so here I just highlight a couple of genes. For example, genes associated with survival as IL-7 receptor, metabolic reprogramming as at L21. So that's very important because I'm going to show you in a few that those cells seems to use now more fat acid oxidation as a memory resource, which is a very is a feature actually of memory T cells or CD8 T cells or T cells recirculation as a CD6012, co-stimulatory molecules, as a CD40 ligand, or important factors, transcription factors, RANKS1 or BLIMP1. Some anti-inflammatory molecules being downregulated as a 10 and a 24. So it was a big amount of genes or the transcriptional network that changed, along with being downregulated. So again, I think, and then I'm going to tell you a little bit in the end of the talk that came from all the studies. BATF3 really regulates a huge amount of genes and make the chromatin accessible for different genes. So anyway, but here we also, as I said, we found that those cells, they have a preference to use OX4s when BATF3 is overexpressed. So, which is again something that memory T cells typically do. All right. So when we saw those cells doing so good in this OT1 system, we thought, well, that's interesting, and maybe it will confer an advantage to those cells in a chronic antigenic environment as a tumor, right? And so back there we used the LCMV virus, the clone 13, as a proxy, and as you know, this induced a chronic infection, and then we decide to challenge those cells in that system. So again, the same thing, we have the wild type, so the control is scramble control of the BATF3 overexpressing cells that got transduced with the virus, with the construct. And then we put those cells in vivo in a fifth into a recipient that got infected for 30 days. So we just prepared the environment for those cells. So then when you transfer the cells at day 30, and then we wait for 7 days for the analysis. Basically, what we checked initially was the numbers. As you know, in that environment, the cells start dying, the cells start getting exhausted, right? So they produce less cytokine, they start expressing some of the exhaustion markers as TIM3, they downregulate TCF1, and then we start looking at the numbers, and this is what we found. We found BATF3, when BATF3 is overexpressed, conferring a big advantage to the CD8 T cells. And then we also checked this data actually didn't published. We didn't really want to mix up because we have an idea to have an odd story. This is unpublished data. So, but we found like TIM3 being downregulated, which is one of the markers of exhaustion. We also have data that I'm not showing you guys today, also not published, where we transfer only the scramble control of the BATF3 overexpressing CD8 T cells, the P14, the ones which are specific for SMV, and then we check for the virus load, and was dramatically low compared to the wild type. But we decided to follow that story, but we didn't have the chance, and then all the effort of the lab moved to another story, and then we have a collaborator working on CAR T cells, and and the life just moved. But as I'm going to show you in few, I think you were back there quite right about it, and it's really nice to see how BATF3 have seen by the field as a potential target for cell therapy. All right, but before that, so everything we got there was done in mouse and in mouse cells, and here we thought, well, that would be interesting to check how it works in human, but of course, in vitro system. And the same idea, so we overexpress BATF3 compared to the scramble control, and that's what we get over time. Again, BATF3 overexpressing cells doing much better. And interestingly, in human system, we saw a slight higher amount of cytokine being produced. So the cells can produce a little bit more of cytokine compared to our control or the empty vector here. So here's just interferon gamma, TNF, and L2. So we thought, well, that seems to be very promising, and we just filed a patent back there in 2021. And speculating that targeting BATF3 or maybe BIM would be a nice way to modify lymphocytes and maybe to make better CAR T cells. And talking about CAR T cells, as you might know, this is one of the most promising strategy to fight cancer. One of them. It is still very complex, and people in the field, they are trying to make it a little easier. And I think it's just a matter of time that you're going to improve this system. But that's the classical system, right? You have a gene for the CAR. As you know, this is a chimeric antigen receptor, so part of, let's say, antibody or TCR. And then you typically use lentivirus to deliver your gene, right? So to get integrated in a genome. And then using that classical system, you cannot really control in which part of the genome, though, that construct will be inserted. And that was a beautiful study where it was a clinical trial where they got very lucky to get, I think, 2 or 3 patients that got 100 that they got cured, 100% remission of the lymphoma. And they followed those patients. I hope and I believe those patients are still alive. That work was published in 2018. And then, so when they got the blood from those patients, they by month number 2, so 2 months later, they realized that half of all T cells from those donors, they are coming from one clone. And then when they gate on the CD8 positive CAR T cells, it's almost 90% coming from one clone. So they got very excited, of course, and when they sequenced those cells, they found that a gene, it's called TACH2, which is a epigenetic regulator, was disrupted. So the insertion of that gene disrupt the TACH2. So then people start, wow, that's wonderful. That's a molecule that you can target to improve CAR T cell. But there are a couple of problems with that because this is a master regulator. But there is still a lot of enthusiasm around TACH2, and I share the same enthusiasm. But what was interesting to see was that another paper in 2023 now showed that because they didn't know how, and then there was a big effort to understand it. And what they realized was when TACH2 is disrupted, or if you remove TACH2 from the system, what you basically get is uncontrolled, or let's say overexpression of BATF3. And they have nice control showing now if you silence BATF3, you basically lose all the advantage that TACH2 disruption was promoting. And then more recently, another study, completely independent, they were looking at, they used the CRISPR screening, the loss of function and gain of function of several transcription factors. They are looking for, factors that could promote an advantage if there is a gain of function or a loss of function for CD8 T cells. In the end, they want to also have some CAR T cell. And then in an unbiased way, they basically found the top hit was BATF3. Again, when BATF3 was overexpressed, they found big advantage in terms of function, survival, memory capacity, fitness, everything. And here they have done several studies. One of them was to check the chromatin accessibility. And here's some regions that got accessible when BATF3 was upregulated or overexpressed. And most of the genes that we also found, L7 receptor, TCF7, which encodes TCF1, a very important gene to be resistant against exhaustion, CD6012. So basically, in those cells, they also switch their metabolic preference from glycolysis to OX4s, but in less extent compared to mice. And one of the problems we have with CAR T cells nowadays is, to make those cells to fight solid tumors. And then in their work, they had a model where they use the CAR T cell and which cannot really respond properly and kill the tumor cells. But when the BATF3 is overexpressed, they have a decrease in a tumor volume. And he's just two different doses, and they speculate that, with that, reprogramming the CAR T cells with gain of function of BATF3, they can use even less cells. And of course, this is still some speculation, but I think we might reach there sooner or later. And what is important to say here is, BATF3, we don't really know what could happen if the cells keep BATF3 unchecked constantly. So we might need to design a CAR T cells where BATF3 will be expressed, but maybe a switch off control should be there too. All right, so that's the take-home message for today. So we know that BATF3 is expressed at the beginning of the priming phase, and it's gone. It has to be silenced. And the deficiency of BATF3 doesn't really impact the expansion of the cells. They expand normally. But what we found was a very aggravated contraction, a lot of genes dysregulated, and one of them, BIM, that's not downregulated, supposed to be, and the cells just contract too much. And what we get in the end is just a small pool of memory cells. But remember, even if you now put the cells one-to-one, they cannot respond so properly. As we learn a little bit from the CAR T cell work, very likely it's also controlling, the capacity of the cells to respond and the quality of the cells in different ways. What we also learn is when you promote the gain of function of BATF3 in CD8 T cells, you improve the memory capacity, the metabolic fitness. BIM is also downregulated, which is good, and then you end up with a bigger memory pool. But more importantly, those cells, they are more resistant to exhaustion, and they are more prone to fight in a chronic antigenic environment. And then with that, I believe that BATF3 has to be more explored in terms of cell therapy to fight cancer. All right, so I just want to thank everybody that helped me with their work, especially Professor Kastenmüller, a spectacular scientist, Carl Commander, a PhD student that I had the honor to supervise back then in Germany, and everybody in Germany that contributed, and the funding that helped us to get that work done. And you guys for listening here for almost an hour. All right, thank you. That's all.
Thanks a lot, Marco, for a great talk. We usually open up for questions from the students first, but I would like to make an exception here because Gustavo is in a rush. So, Gustavo, just fire away, okay?
All right. Thank you very much, Thiago. Marco, wonderful work. It's just one quick question regarding the dynamics, because as you pointed out, BATF3 is a transitory, the expression is very transitory, and BEAM is happening all the time later on. Okay, so I don't believe that BATF3 is controlling directly the expression of BEAM, but you mentioned some epigenetic changes, you mentioned metabolic fitness, whatever. So what is controlling BEAM expression throughout the T cell activation?
Well, yeah, I also share with you the same feeling that it's hard to believe the BATF3 is directly controlling, but probably a lot of things around. Um, so I really don't know. I mean, Beam is up there all the time, and then it goes down when when you have TCR signaling. Um, um, so I believe that cytokines like IL-7 or IL-2 could downregulate BIM, but I'm not 100% sure because BATF3 knockout CD8 T cells, they downregulate those receptors. That might be one of the way that BIM is not put in check, but it does in the wild type cells. But I don't know in the, let's say, homeostatic conditions, how BIM is controlled. What I think, based on our data, is that the TCR signaling is very important and to downregulate BIM temporarily because it also goes back later on.
Yeah, okay. So the quick answer is that it's not known. So you don't
Yeah,
know. Okay, that's very nice because it gives us, ground for investigation. I'm interested in Beam. We have been knockout mice. We've been working a bit with this, and then now we are also doing some stuff with the BATF3 and in vitro activation of CD8, so we can put up like a little
yeah,
project to investigate
nice.
that. And
It's nice
also,
to
we
hear
are
that.
interested in the metabolic modifications during the early T cell activation. So, yeah,
Cool.
let's talk about that later then.
Let's chat. Yes, absolutely, Gustavo.
Okay. Congratulations, man. Thank you so much. Guys,
Thank
have a
you.
wonderful
Bye.
week. Thank you so much. See you, Thiago, again.
Thanks, Gustavo. Okay, we have a couple of questions here from Ruth in the chat. I'm going to read it to you, okay, Marco?
Yeah.
So that was a very informative presentation. And then she wants to know first, how does BAP3 deficiency impact the spatial localization of memory TCD8 cells within the lymphoid or non-lymphoid tissues? And what implications does this have for protection? I don't know if
Oh,
you have
that's a
an
nice
answer to that.
question. I don't
Mhm.
know. I mean, it's funny because I've done this work in a lab that I have the culture of checking the localization of the cells, how the cells behave, and we never test that actually. Um, I don't know. Maybe it will change, but I'm not. I don't know. I think nobody has done this. It's a work to be done, and it's a very very simple question, but I don't know the answer. Never tested.
Okay. And the second question from her is, could engineering dendritic cells to overexpress BAP3 enhance the generation of high-quality protective memory TCD8 cells in vaccine strategies, especially in low immunogenics contexts like tumors or chronic infections?
That's another good question. Well, BATF3 is important for disease development, and it's not known, at least from my knowledge, if you promote gain of function disease, if you get more fit disease, or if they present antigen in a better way. I don't know. I think it's a difficult question to be answered experimentally, to be honest. Um, so because the role that we know for BATF3 in this is based on the importance of BATF3 for those cells to develop, to exist. But well, yeah, so that's something we have to test. We have to promote gain of function, see how the cells, how they behave in vitro in terms of antigen presentation. Why not? But I see, I see BATF3 being targeted in a T cell as a better way than in DCs, especially because DCs, they're replaced more often, right? So every, I don't know, 7 days, something like that. And the BATF3, the T cells, they expand. In theory, they will be forever, right? If they mount a proper memory. I think it's better way to go on T cells than DCs.
Okay. We have questions in Sao Paulo, in Rio.
I guess Miriam was raised her hand before me, right?
Oh, really?
Okay,
Sorry.
thank you.
Sorry, Miriam.
No, that's okay. Diego, do you have any questions from students? Because here is just one question from me.
No, but here is a question for me as well. So
Okay. So Marco, thank you again for giving the talk to us, and congratulations on the data. I wonder, do you have any idea of how these cells that do survive in the absence of BATF3, how they behave? If they have a different antigen, a different repertoire, they would be more likely to become regulatory or...
Yeah, so you're talking about the ones that are still left there, right? They're not yeah.
Exactly. They don't get all extinct, right? There is a subpopulation that does survive. I wonder if you have any information on those cells, because they might exist in vivo, right? They're just a very small population. So I wonder you were able to enrich for them. So
they do. I don't know. I mean, I had the same question. I wanted to follow those cells up. Well, so here's my take. I don't know. I think it's very possible that we have a different clones that they do not need BATF3. The big majority of those cells needs BATF3. I didn't show any data, but we checked all the subsets, the central memory, the effector memory, the tissue resident memory. So it's a very general function, but it's still, to me, as you said, that is a population that requires it. That's my first take. Or what I think is those cells, they are more resistant and they will disappear at the point. But I never follow those, mice to the point to not see cells anymore. So I tend to believe that they exist, but I don't know how they can go through all the process without BATF3. But I think there's a BATF3 independent for sure.
And do you have an idea if the endogenous repertoire, like does BATF3 plays any role in terms of T cell development itself? Is the repertoire intrinsically different, or is only actually important in the secondary response?
No, it's not. Yeah. For example, the BATF3 knockout, I mean, they have the full knockout. They have the CD8 T cell compartment 100% intact. So, well, I didn't formally checked and test for the TCRs if they are different. I'm just assuming they're the same as the wild type, but it's possible. But as I said, in the full knockout, you still can get tons of CD8 T cells, no problem. They develop and they are there. They are naive. You can just for memory information. But that's a good point, good question. Maybe if you do a TCR seq, you might see something completely different, but I don't know. I never did.
Thank you very much again, Marco.
No problem. Thank you.
Okay, so we have 3 questions in line. Diego first.
Okay, so very nice data, Marco. Great talk. My question is not very scientific at all. I would just like to know if is there any kind of pharmacological modulator of BATF3 expression or stabilization or stuff like that that can be, repurposed or I don't know. Do you know something like that?
Well, no, I mean, that is available in the market. I don't believe there is nothing available in the market to maintain BATF3. But first of all, why BATF3 is downregulated? If we understand exactly, because I don't know. I mean, we saw that peak and then it's down. If we better understand how this dynamic is regulated, maybe you can propose. I mean, it'll be hard, okay, because BATF3 is expressed in different cell types. I think we have to have something target specific. But imagine we can propose a small molecule, an inhibitor of I don't know, any regulator that induce BATF3 downregulation, but I think it would be very tricky. And I don't believe enough in that regard really exists. Yeah, so modulating BATF3, I think the best way at the moment, at least in terms of T-cell immune response, is via an engineering T cells, and including the BATF3 information. And as I said, it will be, I think, unsafe if you leave BATF3 unchecked. You have to have a switch control there to switch it off.
All right, thank you.
No problem.
You know, just curiosity if there's something like that. But as
Yeah.
you said, it's hard to know.
Yeah, no,
right.
absolutely, yeah.
All right, thank you.
Okay, Maria Belio.
So, hi, Marco. Congratulations. Nice work. Very nice presentation. So maybe you said it and I missed it, but it's just a very simple question is about CD4 T cells. Do they also express BATF3 around day 3 or something like that? Because we know that TCR signaling is quite similar in CD4 and in CD8 T cells. So I was wondering whether you also have this upregulation of BATF3, and if you looked in the knockouts, what happens with the CD4s?
Yeah, no, that's a good question. Well, we didn't really check CD4s. BATF3 is expressing CD4s too. I was so away from that story for many years, but I think BATF3 also regulates the function of Tregs. So in terms of CD4s, more in the Tregs. And BATF, without the 3, is very important for the CD4 function. I don't know if BATF3 expression in CD4s and related to regulatory T cells follow the same kinetics. I don't know. But this is very simple question. When we are working, sometimes we're so focused on one thing. I could get data on this, right? But I don't know, and I haven't seen data in that regard too. But I know that BATF3 is also important for Treg function, via CD4s.
Yeah,
But
thank you.
That's something you can do maybe easily, yeah.
Yeah, you cannot answer every question at the same time.
Yeah.
Thank you.
All right, Maria, thank you.
Thank you, Maria. Fernando.
Hi, Marco. Very nice work. Very nice talk as well. So I really like to study dendritic cells. So my questions, they are from the DC point of view.
Okay.
So from the BATF3 mice, so CDC1s are usually back after infections because of interferon type 1 production. Have you checked in your model if they are really back or if they have a function after the infection?
Well, so you mean the DCs?
The CDC1 specifically.
CDC1, yeah. The system we used, we basically have the DCs functioning all the time. We didn't check anything from the GC side because we always have the wild type mice as a recipient, and we just transferred the CD8 knockout. The only knockout compartment was the CD8 T cells. So we just assumed that the DCs, they were just functional and they were there at the moment they're supposed to be. But so They were probably there all the time. But as I said, we didn't really follow any aspect of DC function because, we just transfer the CD8 T cell KO into the recipient.
I see. So I was
Yeah.
also wondering about like the T cell interaction with the CDC2s, because their CDC2s are independent of BATF3, and they are usually not really good in CD8 T cell, more related with CD4. So I was wondering if you have like any information about them.
Yeah, no, as initially introduced, those studies, you might know those from 2015 and 17 from Miller group. So it seemed the CDC2s in that moment, they are just instructing the CD4s, while the CDC1s are instructing
Chegou!
CD8 T cells.
Não.
And then, wait, something, no. And there is a noise there. All right, now it's gone. All right, so it was a delay here. So yeah, we didn't really evaluate any role of the CDC2s on CD8 T cells, and we didn't really check any spatial distribution of those cells and see with which population they are interacting with. But what I believe is, the CDC1s, they were there and they were doing the antigen presentation to CD8 T cells. And then, in that later phase of the priming by day 3, they also work as a platform. I mean, that's the moment the CD4 and CD8 meet, and they are seeing the same antigen. But as you're right, so CDC1, CDC2s, I think they're important to those different type of T cells, CD4 and CD8, in the very beginning, maybe the first 24 hours, but later on, those cells, they encounter the XCL1 disease to meet. And, the deficiency in BATF3 and CD8 T cells, specifically, I don't know how much changed those interactions. We didn't really check that part.
I see.
But
Thank you.
It's possible, yeah.
Yeah.
Any other comments or questions for Marco? Marco, I think that's it. Thanks a lot again for being around. Yeah, and we're excited for your work now at Ribeirão. Hope to meet you soon there.
Yeah, thank you, Thiago, for the invite. As I said, it was great to talk to this audience again and share the sort of BATF3. I hope we can hear more about that molecule being into the clinics and helping patients, especially in Brazil, via SUS.
Yes, very nice. And that's all, guys. See you all next week for our last edition of this semester of the Immuno webinars. Okay. Bye-bye. Thank you.
Falou, tchau, tchau.